In this set of 3 lectures, we'll talk about development, fluctuation, and adaptation of populations. We'll begin with development. After this lecture, you'll be able to identify major events that occur in the development of rumen microbes, beginning from the birth of the host. The textbook does not cover this subject, and so there is no reading from it. To give an overview of what we'll see, after birth, the rumen is rapidly colonized by bacteria and methanogens. In the following weeks, the fungi and protozoa colonize, and the abundance and diversity of microbial groups increases. In the following months, abundance and diversity reaches that of the mature rumen. The following slides will present a timeline of development. Most data refers to calves in a traditional feeding program, that is, calves that are given colostrum at birth, a combination of milk and starter at young age, and then weaned at several weeks of age. Thus, the changes that we see in development are confounded with changes in diet and management. Immediately after birth, the gut is initially sterile, but colonization occurs within minutes. In the timeline below, you see this signified by the arrow with initial colonization pointing at 0 weeks. That is, it occurs within just a few minutes. The first few days after birth, methanogens and major bacterial groups colonize, but the rumen is still far from mature. So we see down below that anaerobes are already high, at concentrations of 10 to the 9 per milliliter. Facultative anaerobes are also high, at concentrations of 10 to the 8th per milliliter. Methanogens are present, all the major substrate degrading groups are present, the diversity is low, and taxa are different from the mature animal. In subsequent slides, we'll see data that support these points. This study looked at changes in bacteria and methanogens over several weeks after birth. These two figures show the change in facultative bacteria and anaerobic bacteria. We see values for both conventionally and early weaned animals. We won't concern ourselves with differences between conventionally and early weaned animals, we'll just look at the general patterns. At 0.5 weeks of age, or the first few days after birth, we see that facultative bacteria are relatively high, compared particularly to values later after birth. The anaerobic bacteria are rather high, at 10 to the 9th per gram, but they'll be higher still in subsequent weeks. We see here similar figures, but for proteolytic bacteria, lactate-utilizing bacteria, and amylolytic bacteria. These were grown on selective media. We see each of the groups is present, but at relatively low concentrations compared to values later after birth. The methanogens were also quantified, as well as the cellulitic bacteria, and we see again that these are present, but at relatively low concentrations. This study used 16S rRNA gene sequencing to examine the diversity of rumen bacteria in cohorts of animals from just a few days of age up to 2 years. This table reports a number of OTUs, the operational taxonomic units. These are roughly equivalent to species. We see that at 1 to 3 days of age, there are several hundred OTUs, but that still pales in comparison to the number of OTUs for older animals. This is a Venn diagram from this same study that shows the number of shared genera by age. We see the ellipse for animals for 1 day of age is pretty distinct from ellipses for animals of older ages. By 3 days, more genera are shared with the mature animals of 2 years of age. 1 to 3 weeks after birth, we see colonization of the fungi and the protozoa. The fungi will colonize as early as 8 to 10 days after birth. The protozoa will colonize as early as 15 to 25 days after birth. At 3 weeks after birth, anaerobes will plateau to a concentration of the mature animal, but some differences still remain. First, the abundance of most functional groups is high, but cellulytics and methanogens are still low. And second, the taxa are still different from a mature animal. So to go to the timeline below, we see that anaerobes plateau at concentrations of 10 to the 10 per milliliter. Facultative anaerobes are declining, functional groups increase, but the bacterial taxa are still different from the mature animal. Going back to the figures that we saw earlier, we see by 3 weeks of age the facultative bacteria are indeed declining, and the anaerobic bacteria have nearly plateaued at 10 to the 10th per gram. The concentrations of the proteolytic, lactate-utilizing, and amylytic bacteria have reached pretty high concentrations by 3 weeks. However, the concentration of the methanogens and the cellulytics remain low. 6 to 12 weeks after birth, the trends established earlier continue. The facultative anaerobes decrease, the cellulytics and methanogens continue to increase, and the abundance and diversity of normal taxa increases. We see in the timeline below that by 6 weeks, some bacterial taxa are similar to the mature animal, and we see between 9 to 12 weeks, the facultative anaerobes have reached a low concentration of 10 to the 5th. Also, bacterial taxa are becoming more similar to the mature animal. Referring back to these figures once again, we see facultative bacteria have reached a low concentration, and the anaerobic bacteria remain high. The proteolytic, lactate-utilizing, and amylytic bacteria still remain high, and the methanogens and cellulytics are finally reaching high concentrations. We see from the sequencing study that by 2 months of age, the number of OTUs has risen, but it's still much lower than that of a 2-year animal. We can see from the Venn diagram that more taxa are shared between the 2-year animal and the 2-month-old animal than animals of younger ages. At 6 months after birth, the rumen is nearly that of a mature animal. By 24 months, we presume the animal is mature. We see that the number of OTUs has nearly plateaued by 6 months of age. And we see that there are a number of shared genera between the animals of 2 years and 6 months of age. We've mentioned the times at which the rumen is colonized, but we haven't yet discussed the mechanisms. Typically, colonization occurs by contact with a mature animal's saliva by licking or feeding. Copophagy can be another mechanism of colonization. If the animal is in isolation, contact with other animals like birds or rodents is likely to affect colonization. An old culture-based study from 1960 established that. It also showed feed is an unlikely source. It is almost impossible to prevent colonization of bacteria. Germ-free ruminants can be generated if they're delivered by hysterectomy or C-section and then raised in sterile isolators. You can see an image of a sterile isolator to the right. As we'll discuss later in this class, germ-free and non-abiotic ruminants are important models in studying microbiological function. Colonization of protozoa can be prevented if an animal is raised in relative isolation from other animals. Preventing this colonization is one method of studying protozoal function. In an earlier lecture, we referenced removing protozoa from the rumen, and we can do that by emptying the rumen, washing the walls, starving the animal, and then allowing the rumen to be recolonized with bacteria. But isolating the animal is another method by which we can prepare protozoa-free animals. These show isolation pens for defaunated animals. You can see the high walls to the left and to the right. You can see that food and water is given to each animal separately. This prevents recolonization. We can prevent colonization by fungi as well, but it's more difficult than with protozoa. Colonization can occur over long distances by aerosols. Viable fungi have been found in dried feces. To this point, we haven't talked about the host, but the host can strongly control the composition of the microbial community that develops. One study took communities from 2 cows and swapped the rumen contents of these cows, and it found that the communities returned to their original composition even after swapping the rumen contents. In the figure at right, you can see several nodes representing days after swapping rumen contents for one cow. The closer the nodes, the more similar the composition is. We can see the change that occurs when the contents were swapped. We can look at the difference between the nodes labeled pre and post in reference to the time after swapping. We can subsequently follow the red arrows at several times after swapping. We can see at the end, at 65 days, that the composition has returned close to that what it was pre-swapping. The mechanism is still being elucidated. This experiment was replicated with 2 more cows, and in that case, the composition did not return to the original pre-swapping state. We'll have to be able to repeat the phenomenon more consistently before we can elucidate the mechanism. The take-home messages from this lecture is that the gut is initially sterile, but colonization occurs within minutes. The protozoa and fungi do not colonize if the animal is well isolated. The rumen takes months to reach peak diversity and to become mature. The host is important in determining the composition of the bacteria, but mechanisms require more study. In this set of